Hello, thank you so much for joining me. Today I'm going to do a presentation and I'm going to give an overview on the requirements for applications for biological and biotechnological medicinal products. So I'll talk about the regulatory requirements and the scientific aspects. This is going to be one of a series of presentations that I'm doing, and I'm basically combining what I've worked on in the past and on some new material that I've read and that I've learned from studying the legislation and from the various courses that I've gone on and reading the regulatory guidelines. So this is an introduction video. So as I said, this is going to be a series of videos, and in the first set of videos I'm going to talk about or in this first video I'm going to talk about a theoretical approach, if you like, for a protein, and I'm going to call this a pegylated monoclonal antibody fragment that's used to treat an autoimmune disease. So this kind of is an overview, if you like, a regulatory flow chart overview, which details the process, if you like, the life cycle of the cells that ultimately produce the protein product, which is the antibody fragment. So you start off with primary cells, and then these primary cells then are used to form the parental cell line. Now, primary cells are usually obtained from tissues or organs, and these tissues or organs have to be, I don't know, they have to be obtained, if you like, in a, following the relevant regional legislation or guidelines or laws. Now, this is I've put under in that square bracket, or in that square shape, good tissue practice, and I will be going through that in a second video because I don't think I'll have enough time in this video. But basically, these primary cells have to be obtained in a manner that is traceable, that is, if they're human beings that have donated those cells, that we've got to take into consideration the health of the human being, the region, the age of the human being, because that may impact on the the life span of the cells that we used if we're not going to immortalize those cells. So also, the donation of the cells have to comply with the various laws with regards to the General Data Protection Regulation in the European Union, and also with regards to any confidentiality requirements. I think it's called the HIPAA requirements in the United States. So good tissue practice has to comply with a whole host of requirements, and that will be something that I'll cover in another presentation. But if you have your immortalized parental cell line, say for example in this theoretical example that I'm providing, those parental cell lines, we normally consider those to be, like for example, if we're talking about a cell that's going to be transfected with a particular plasmid, then the parental cell line, these are the untransfected cells. So a cell has been isolated, if you like, and selected. A cell clone has been selected, and that particular cell line has been, or that cell has been, expanded. You have a population of those cells, and those cells can now be considered to be the parental cell line. Now, when we transfect the cells in this particular example that I'm giving, to be able to express the particular genetic sequence that will yield the antibody fragment, when we transfect those cells, those are what we refer to as the cell substrates, because the cell substrates are the cells that can produce the protein of interest when they are induced to do so. But we have to consider other things. So we have to consider how we are going to manufacture. We will have a cell bank, if you like, a master cell bank and a working cell bank, and I'll go into more detail of what these actually are. But in a nutshell, they're just a uniform aliquot from a single pool source of cells. And they're basically the master cell bank is the initial, if you like, the first step, and the working cell bank is derived from the master cell bank, and then the working cell bank is used to produce more cells, to expand it further until you get to the what we call the end of production, or the limit for in vitro cell age for production, where this is the volume of cells that is required to produce a good yield of the protein product once we induce those cells to do so. So the working cell bank and the master cell bank and the cells at the end of the production of the cell substrate at that particular stage, that I don't know, that period, or that manufacturing process has to be characterized. The master cell bank has to be characterized, and it has to be predefined specification. It also has to be characterized in terms of its viral characterization. The working cell bank usually undergoes some qualification, but not full qualification in the same way as the master cell bank, either one or the other sometimes. And then the limit for in vitro cell age for production, that period of time from the thawing of your master cell bank to the limit for your in vitro cell age, that whole period, and that whole all of the cell expansions and cell culturing, etc., that occurs, you have to be able to show that there is no impact on the quality of those cells in terms of their viability, in terms of their ability to produce the protein of the quality that is expected for the intended use. So all of that, from master cell bank right through to the limit for in vitro cell age, has to be qualified. You have to show that the quality of the protein produced at the end of the production, if you like, or at the limit for in vitro cell age, that quality is equivalent to, I don't know whether this is always possible, but equivalent to the protein that would be produced from the master cell banks. Yeah, and so therefore you've qualified and you've shown that there's no impact on your manufacturing process where you increase your volume of your cells, that there's no impact on the quality of the protein that you're producing. Anyway, so your production would be where you would then harvest your proteins, and you would then, concentrate them, purify them, and then further manufacturing. In this particular example is the pegylation step, where you would then do the reaction of the antibody with and form the pegylated antibody fragment. So this is kind of how I picture it in my mind. Now, as I say, I've started with the primary cells, but in this particular case, it's actually a parental cell line, the Chinese hamster ovary cells, which is a well-established and well-recognized and well-characterized cell line. So we don't really have this primary cells. But if it were a new product or some other product, you may start with a primary cell. Yeah, so you start with in my mind, I kind of think of it as you start with your primary cells, then you create your parental cell line, and then you have your master cell banks, your working cell banks, through to the production, of the end of the production, the number of passages that you may need to do in order to obtain a good volume of cells that can produce a good yield of your protein product. Yeah, and once you do that, the cells are induced to produce the proteins. I'll go through that, and then the proteins are harvested, purified, concentrated, and then treated to a further chemical reaction, and this is the pegylation. And then in this particular situation, in this particular case, they produced the bulk product, which was then frozen and stored until it was required. And when it was required, the finished product would be manufactured by thawing the bulk and automatically or automating the vialing of the bulk into individual vials of a certain aliquoted volume before it would then undergo further testing for release into the clinical trials. So this is just an overview, and I talked about the regulatory aspects, what you need to be thinking about, and what information you need to be providing. Right, so these are the ICH guidelines that I sort of had a study. I did a good study on these guidelines, and these guidelines are very thorough, I think, very readable. I'm not going to go through all of it, but I will go through some of the information that I found to be particularly interesting and useful. And I'll be going through this information throughout the series of presentations that I put together. So this particular slide, I was just thinking about the scientific aspects as well. So I just added some additional information that you would need to consider. So for example, I think I've already talked about the fact that the master cell bank has to be fully characterized. You have to provide defined preservation conditions and techniques. So these are the scientific things that you need to sort of nail down and really fully understand what kind of preservation media you're going to be using that's going to produce the best viable cells. You know, what kind of thaw conditions, how do you thaw your conditions, how many vials are you going to use or containers are you going to use for your master cell bank in order to create your working cell bank. And of course your viral characterization. So your critical quality attributes, you have to understand that at this stage, and you also have to understand your critical process parameters, because you're thinking about your manufacturing your manufacturing process, and your manufacturing process will involve you manipulating or at least handling these cells. So you have to understand what processes or what actions or what unit operations could potentially have an impact on the quality of your product, and therefore you need to monitor and tie down those unit operations that potentially could impact on the quality, the critical quality attributes, and therefore having that understanding and understanding what steps need to be monitored and controlled means that you have a control strategy in place in order to be able to produce a product that is of the required quality for its intended use. So all of these things need to be considered from a scientific perspective. Purification, concentration, and your characterization again. What techniques can you use to purify your product from a scientific perspective? What techniques do you use to concentrate your product? How does this impact in terms of the quality of your material? So, as I said, all of these things are important. The other thing, as I think I alluded to in the previous slide, was that for this particular example, we used the Chinese hamster ovary cells, and these were transfected with so that the Chinese hamster ovary cells were the parental cell line, and they were the untransfected recipient cells, but we transfected them with these plasmids which were grown in these E. coli systems. And actually these E. coli and the plasmids were, I believe they had cell banks for them as well. And your master cell bank, working cell bank, I think I've talked about all of this. yeah, I think it's very similar to the regulatory aspects, but just some additional things to consider. So you're in this particular thing. Began with an overview of the manufacturing um, process, and as I said, the parental cell line is derived from the Chinese hamster ovary cells, and those cells are specifically chosen because they were or are able to produce complex proteins with human-like glycosylation patterns. Now, glycosylation is one of these post-translational modifications, and the post-translational modification, that happens for proteins. I mean, I'm a chemist, so my background is mainly chemistry, but I think I do remember that. And you want to have them human-like to reduce the potential for inadvertent adverse reactions. I presume that would be the reason why you would do that, and also for the efficacy of the product. So your master cell bank and your working cell bank are established. And from there, you then expand your cells, and then you move on to your protein expression. Right, so I think I've talked about this really in the previous slide, where I've talked about the Chinese hamster ovary cells are the parental cell lines that are transfected with plasmids, and they contain so the plasmids contain the relevant genetic information for the humanized fragmented antibody. Yeah, and the cell substrate. So when you read the guidelines, the ICH Q5, I think it's Q5D and Q5C, I can't remember which one, it could be D and E. They do talk about cell substrates, and what these are the cells that actually produce the protein of interest. And the master cell bank and the working cell bank can produce the cell. They are cell substrates. They can produce the protein of interest, but really they're the kind of like the starting point in terms of our manufacturing strategy, where we can, always have a common source, the same source of cells to manufacture the product, and from doing that, it's a risk based approach, if you like, reducing the risk of variability, because you're always using from that same source. And we also think long term. So we think in the future, how much of this product do we intend to manufacture? We need to think about that right at the start of our development, so that when we are laying down and establishing our master cell banks, we can establish enough material that will last for the lifetime of this product, right from the clinical trials, right through to the marketing authorization, right through to post-marketing authorization, when the product is being marketed, not just in the country of interest or the region of interest, but maybe even worldwide. So we need to think strategically and long term, and we need to think about all of the regulatory requirements for all of those countries in all of those regions, because they're all different, even though they have the same scientific justification and rationale. They are all different, but we do need to think about it so that we cover all of the requirements for all of the countries that we plan to market in. And we make that consideration at the start when we're actually choosing our primary cells, and we're choosing our parental cell line. So I don't know, I think I've gone off target. I've sort of digressed a little bit, but these plasmids that are used for transfection, they were grown and amplified in the E. coli systems, which were also banked and appropriately characterized. And the master cell bank and the working cell banks, they are established from the parental Chinese hamster ovary cell, the CHO cell line. And these are what we have to consider, these cell banks that we have to consider and plan in advance, long term. So we have to really think strategically. So I talked about the protein, because at the end of the day, that's what this is all about. This manufacturing process is to produce a protein, and the protein is being produced by the cells. So the cells, if you like, they're like the production engine, if you like, that produces, or the production line that produces the product of interest. And these proteins are produced by the expression of the genetic construct, which was within the plasmids, they're contained by the plasmids. So the protein expression, so at the limit for in vitro cell age for production, i.e. when you have enough cells to produce a good yield of the protein, the CHO cells are then induced to express the desired humanized monoclonal antibody fragment. This is typically achieved using a strong promoter system or inducible systems such as doxycycline and tetracycline responsive promoters. Now, I will go into that in detail and just explain what I'm talking about there. But basically, you induce these cell expression, the expression of these genes to produce the proteins, and you do so in these particular environments. So using these doxycycline and tetracycline environment, the promoters that are within this genetic construct respond to that particular environment and are likely to produce the protein of interest. Once you've produced enough protein, you then harvest and purify the protein using various methods that I will touch on briefly, but I'm not really gonna, this is the theoretical, I've left out a lot of details, but I'll give an overview of the ways that you can harvest and purify your protein. And then this particular protein was then chemically modified to form the pegylated, humanized, if you like, monoclonal antibody fragment. And these particular fragments were used to treat autoimmune diseases such as rheumatoid arthritis and Crohn's disease in the clinical trials. So that's kind of, that's kind of the theoretical sort of process that, that I was looking at. So let's talk about establishing the cell bank. The master cell bank and the working cell bank. So as I said, the master cell bank is created by amplifying a single CHO cell with a stable gene expression for the humanized monoclonal antibody fragment. Basically, your CHO cell is transfected with the stable gene that can express the humanized monoclonal antibody fragment. Your working cell bank is derived from your master cell bank. So your master cell bank, as I said, is from this pool of a single pool of cells, that have been so that it's uniform, and then from there you then expand to produce your working cell bank. So your working cell bank is derived from your master cell bank. You have to characterize your cell bank, and you do so with pre-specified or predefined specifications that are relevant for the cells themselves, and relevant, and you link it to the protein, the quality of the protein for its intended use in the clinic. So ultimately, your characterization is ultimately linked to the efficacy, i.e. how it works in humans. So ICHQ5B talks about the quality of the biotechnological products, and these are the key tests that it talks about. So this is the genetic stability testing. So this is basically one of them, I can't remember which one it is, where we show that our production process, where we have done multiple passages, doesn't impact, you don't get mutations or you don't get changes in your genetic sequence of interest, the transgene, if you like. You don't get any changes in that it remains stable. And you do that using a number of tests, and that guideline will provide you with the sort of test like next generation sequencing, say for example, or PCR. You know, so you use those to show that basically you qualify in your manufacturing process or your process for producing your cells. You will then you know, show that your genetic sequence has you know, to a large degree remained unchanged. Obviously you may get things like variants that don't, you know, that don't change, but the guideline gives you you know, information with regard to that. And then you'll be doing things like microbial testing, so you'll be doing sterility, bioburden, and mycoplasma testing, and this is to, you know, to show that there is no, or well, you have a strategy and you have to characterize your whole manufacturing process for your viruses. Yeah, so but you have to do, as per most of the regions, European, US and Japanese, all of these Greek countries, Chinese, they all have sterility, bioburden, mycoplasma, and you'll be looking at your adventitious viruses, just to show that your product is safe and efficacious. Well, safe because of the nature of the products. They are derived from biological materials where you have the potential to have adventitious agents or endogenous viruses that could potentially cause serious adverse reactions in patients. So you need to sort of put in a risk a strategy for that. The other thing that you have there's growth, kinetics, and productivity, established cell viability and protein yield over time. So again, we've talked about that with regards to qualifying that whole process, and your cell line identity, and that's basically confirming. You always have to do an identification test to show that what you have or is representative of what you actually expect it to be, and that it's nothing else. Yeah, so this is through short tandem repeat profiling is one way of doing that. Right, so as I said, at the defined, whatever that defined in vitro cell age, or the limit for in vitro cell age is, you will then induce those cells to produce the humanized monoclonal antibody fragment. Yeah, and the induction, well, first of all, the promoter, as I mean, you have to sort of there's loads of videos out on YouTube on you know, the various parts of the gene, and the promoter is the part that, you know, it triggers, if you like, the expression of that gene and the production of the protein, ultimate production of the protein. Yeah, so that you have to, if you haven't got a background in that, you have to do the research for that. But the promoter is a viral promoter like cytomegalovirus, and this is a very common promoter. It's a type of, I think it's a type of herpes virus. Oh, I'm going to sneeze. It's a type of herpes virus, and this viral or cytomegalovirus promoter, CMV promoter, I mean, it is widely used, and it's derived from an immediate early gene of the human cytomegalovirus. Yeah, and in terms of, it's a regulatory element of the gene. Yeah, and it really drives the strong constitutive expression of the gene in mammalian cells. So when the conditions are ideal, you will get the strong expression of that particular gene to produce the protein in the mammalian cells. For this particular system, doxycycline was the activator of the transgene in a tetracycline control system. Remember, this is my theoretical approach. It's not I can't say what was actually used in the real world when we did the actual thing, because it would be confidential, and in any case I can't even remember because it was a long time ago. So here's how it works. So you have your regulatory element, that's the promoter, and there's some other things in the gene construct as well. And the gene of interest is placed under the control of a regulatory element that can respond to specific signals, like the presence of a drug or a change in the temperature. External triggers. So these are kind of like how these kind of promoters work. Yeah. So doxycycline can act as a trigger. In tetracycline controlled inducible systems, the presence of doxycycline binds to a specific regulatory protein, yeah, which then activates the gene, leading to the protein production. So that's how it actually works in the cells. Do you know what I mean? So the protein will be in the cell. The doxycycline binds to that protein and triggers the gene to activate and to start to express the protein. And the advantages is that these systems offer precision, allowing manufacturers to prevent unintended protein production, yeah, which is what you want, that could harm the cells or reduce efficiency. So you want to be able to control when you produce your proteins, and that's one of the advantages for that. They also ensure protein production begins only when the cells are at the ideal stage, yeah. So I don't know, maybe what I need to explain is what are inducible systems, I guess. Um, they're kind of like, I think from that explanation, they're like mechanisms that allow you to control when the gene is activated or when it's turned on. And basically it means that, you know, you can control when protein production occurs, and you don't want to have unnecessary expression before the optimal time. So you want to get the expression at the end of the process where you've produced your cells, not before. So you can grow all your cells first and then place them in the correct environment when you want to start the protein production of interest, kind of in simple terms. Now, I just thought I'd put a little bit of something with regards to contamination, because this is something you always have to do your risk assessment, and you have to think about contamination, because if you're using a doxycycline and tetracycline responsive promoters, then that could potentially mask bacterial contamination, yeah? And so how do you account for that? Do you know what I mean? And I guess one way of accounting for it is to have your processes in place to reduce the potential for bacterial contamination, because you don't know whether or not that would have occurred or that has already occurred. You don't know if you've got issues. So you would therefore be looking at your raw material, you're looking at your suppliers and looking at the tests that they do, and having measures in place with your third party providers to ensure that they do thorough testing of their raw materials, so that you don't have the contamination of your raw materials, and also what kind of testings you would do, say for example with raw material suppliers. Also with regards to the raw material itself, can you replace material that could potentially have certain bacteria associated with them, like for example materials of human or animal use, with synthetic materials? Is that going to impact on the quality of your the viability of your cells and the quality of the protein that they produce? You have to think about all of these things. You have to think about your environmental monitoring and maintaining your clean room. You have to think about your in-process testing, regular testing, to ensure that your cultures remain uncontaminated. And you have to think about cross-contamination as well. And as I said, high purity reagents, increases the or reduces the chances of contamination. So all of these things you have to think about. You take a risk based approach based upon a risk assessment, based upon putting in place risk control measures to mitigate potential contamination due to raw materials. I think I already talked about the cytomegalovirus promoter, so I don't really want to spend too much time looking at this, but you can read this at your own leisure. But as I said, it's the regulatory part of the genetic construct, and it's responsible for the expression, and it can be tightly controlled. And it's compatible with most transfection systems. Yeah. Yes. So I talked about purification. So after harvesting, your protein will undergo some robust purification process, and this is going to be in compliance with. I don't know whether it would. Well, you would need to undergo robust purification. You want to get rid of any impurities that are as a result of your process. So your process impurities, any potential impurities that are endogenous within your product. You want to make sure that you have a highly pure product as possible. So you would, purify, and this is removing the host cell, removing the cells themselves, and making sure that you have a manufacturing process to do that. So chromatographic methods are something that is routinely used. Ion exchange chromatography, size exclusion chromatography, and also your filtration. So there's ultrafiltration and diafiltration. Ultrafiltration is more about concentrating the protein, and diafiltration is about also concentrating the protein, but replacing the buffers with other, or replacing buffers by continuous filtration. So it's like a buffer exchange, and it can be useful for removing low molecular weight impurities. When you have or when you exchange buffers, you can actually inactivate certain viruses, and of course you do need to validate and describe that as part of your characterization in terms of your viral characterization. But you just bear that in mind. Yeah, and so your viral clearance validation, again, this is critical for your product safety. And with regards to ICHQ5A, you want to be looking at the potential sources of viral contamination. You want to look at your cell line qualification. You want to look at your testing for viruses in the unprocessed bulk, and your action plan for viral clearance. You need to be able to validate your viral clearances, or at least qualify it or characterize it. And the ICHQ5A has given, theoretical examples. So in the theoretical example, in the one that I'm providing, this will be characterization of your viral clearance procedures. Some of the examples you can use is low pH a low pH can inactivate some viruses. So some viruses are deactivated by exposure to low pH conditions. Nanofiltration again uses filters with pore sizes that are small enough to remove viral particles without affecting the protein. I must admit, with regards to that, I did obtain that from literature sources, so I'm not familiar with nanofiltration, but there it is. Right, so that's the end of this video. In the next video, I will be talking about, or in the next, the next video, I will be talking about regulatory guidelines, the legislation that you need to follow, and the relevant regional legislation, regional laws, and regional requirements. I hope you've enjoyed this video. If you have, please give it a like. Please consider subscribing. Thank you so much. Bye bye.